Good morning, biology students. It's me, Mr. Cheney, with another brilliant tutorial. Today, it is a science skills activity from the eText. This one's called Cell Transport in Plants. So you'll see in Google Classroom that it has been assigned to you. And you click on this link, it's going to take you directly to the textbook. It's kind of like a lab, and so here's how it works. Once you get there, it's going to look like this. And you're going to want to read through things, so I'll read some of it here since we're doing the tutorial part of things. So once this loads up, I can do so. So here's the investigation. Happy Acres Farm was once a highly productive bean farm, but crop yields have been decreasing over the past few years. The farm owner noticed her plants looked water stressed, even though she was sure they were receiving enough water. In a few places, with particularly noticeable bare spots, a white powder was seen on the soil. Looking to find out why, the farm owner had the soil tested. The problem seemed to be salinization, or high salt content in the soil. So clicking on the magnifying glasses to learn more about the problem, then click Next. So you'll need to click on these and read through them. So inefficient irrigation or over-irrigation can increase soil salinity. Excess water dissolves salt from bedrock in soil. So excess water dissolves salt from bedrock in soil. As the water evaporates, the salt is left on the soil surface. Some sources of irrigation water already have a high salt content. When the water not used by the plants evaporates, this salt is left on the soil. So I'll close that and I'll read this one. Salinization of soil is not a small problem. The United Nations estimates that 50% of world cropland is salt stressed, and this number is increasing by 1 to 2% yearly. However, all plants need some salt available to grow. Last thing to read right here, high salt content works against plants in two ways. Sodium itself is toxic to plants at high concentrations. It disrupts photosynthesis, protein synthesis, and lipid metabolism. And number two, high salt concentrations in the soil change the osmotic gradient in the plant roots, making it difficult for plants to absorb enough water. Okay, so once you've read those, you're going to get this next button here, and here's really the crux of the experiment. So to look at the effect of salt on bean growth and crop yield, you grow 4 sets of pinto bean plants in soil watered with different concentrations of salt solution. The measurement, ds/m, refers to the electrical conductivity of the solution. So the bean seeds are allowed to germinate before they are exposed to experimental conditions. After 90 days, the plants are harvested. Weigh a plant grown at each salt concentration by moving the plant to the scale, then count the number of pea pods on each plant and find the mass of the beans harvested. After you record your data in your notebook, click Next. So here's what we do. We're going to start with a 0 salt concentration. We'll weigh it, 18 grams. Oops. And we will click on our notebook here. And 0 is 18. And we're going to do that for each one of these. So you'll take that off, put that on, and so forth. I'm not going to put all the answers for you. I'm just going to fill this in with a random number so I can show you the next part. But you're doing the whole plant, and then you go to the bean pods and you count those. So for 0 salt, you count these 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So that one's 17 for the pods, and then mass of beans. So we need to go to mass of beans. The 0 salt's right there. That's 36 grams. So row number 1, I've done for you. You're going to do the next 3 rows. And again, the textbook's going to grade all this, so you do want to get the correct answer. So once you've got all those numbers in, you're going to get another Next button. And now the last part of this is salt-tolerant species. So due to the change in soil salinity at Happy Acres, the farm owner has decided that she will need to shift her crops to more salt-tolerant varieties. Some salt-tolerant species seem to have levels of the amino acid proline. Proline may help maintain the osmotic balance between the soil and roots. Click on the salt-sensitive and salt-resistant labels under the cells to see how the 2 root cells are affected by pure water and a 10% salt solution. Then answer the question in your notebook. So if you click on here, You can see that H2O goes into the cell when we're talking about distilled water in a salt-sensitive plant. And when we click on salt resistant, H2O comes in, but it also goes out. So that's why you have plants that aren't going to look so good because of the salt solution. So if you go to our notebook and you answer this question, then you'll get to submit your lab. So one of the symptoms of high salt concentrations in soil is plants that appear dehydrated even when they are receiving adequate water. So how can you explain this phenomenon using what you observed in the root cells of the salt-sensitive plants? So salt-sensitive, you want to look at what the reaction is here, and then you'll answer that question. Once you have an answer, and you'll have the submit lab, and I will see it in Google Classroom with a score. And that's it. That is the lab for today. If you need help during the lab, I will be in class. And I will see you back in class.